So I first wanted to welcome Dr. Fabian Schroeder from Echo Refugium, who's going to be speaking about DNAByte, a modular framework for the simulation and analysis of DNA-based data storage.
Good morning, everyone. Thank you for the introduction. Yes, I will be talking about simulation today, why we need to simulate data storage, how we could simulate data storage, and I would like to introduce DNAByte, which is a framework that we've been working on for the last 1.5 years. Okay, so my talk is structured as follows. I will sort of introduce our project very briefly in the beginning to give you an impression of what the problems are that we are facing and where we come from. And these problems should then motivate our simulation approaches. I will then switch to giving you an overview of the status quo of DNA data storage simulation tools. Then I will introduce our framework and conclude with a brief discussion. Okay, so my company is one of the lucky ones that are participating in the DNA challenge, a four-year project on DNA data storage. And our project, to be more specific, deals with developing strategies for in vivo-based data storage. Okay, so we have two consortial partners, the AIT and TU Munich, who are developing strategies to use bacteria to produce here. I don't know how this is not working. To produce short single-stranded nucleotides, and these nucleotides will serve as building blocks, which we then use to assemble by hybridizing and ligating and to encode information. And at the very end, we want to integrate this data into cells. And so this is the framework of our project, and Echo Refugium is in charge of developing coding strategies and simulating this project. Okay, so when we, at the beginning of the project, conceptualized our work and with talking about the advantages and disadvantages, we identified the advantages and disadvantages that we would like to study further. And I would like to point out two of them: the lower data density, that is one of the main disadvantages of this approach, and the inherent error correction, because these are the two. Yes, if that's okay. So these serve as good examples for how we use simulation in this project. Okay, so data density is one of the most widely used metrics in encoding. So when you develop an encoding scheme and you want to present the advantages and disadvantages or the benefits, very often they talk about data density. And I will not use this. So in our project, obviously we can calculate the metric. We can the data density is just the base 2 log of our library divided by the length of the oligos that we use to assemble the information, and we can plug in the numbers and we can get a value, and then we can compare it to other encoding methods. However, we at the beginning reached the conclusion that this metric is not a good way to really compare encoding methods. If you think about comparing two completely different decoding approaches, one, for instance, based on de novo synthesis, where the information really lies in the sequence of nucleotides, and one where your information consists of building blocks, of motifs, or whatever you want to call them, the data is represented in a completely different way. So you might have longer code words, you might have more repeating motives, you might have problems with CG balance, etc. So the data representation will also affect the errors that will be produced by your encoding approach. And these errors, of course, then lead to the necessity to include error correction and also to the amount of error correction that is necessary to be able to retrieve your data. So the metric density is a very limited view of your encoding scheme because it doesn't account for the real characteristics of your encoding strategy in the sense that how it creates errors for synthesis, storage, and sequencing. So we switched to analyzing our data density by what we call benchmarking. So we specify a use case and sort of obtain our density by means of simulation. And I think you can get a much more reasonable view or a better impression of what your encoding what the density of your encoding actually is. A second example is the inherent error correction characteristics of your encoding method. Now, if you encode data by assembling block by block, you do not use the entire sequence space. So essentially, the information that you encode is not not element of the set of all possible combinations of A, C, G, and T, but you work in a very reduced sequence space. And this can be used for error correction. You can, once you sequence the data, of course, you can use this, you can be projected into your subspace and you can detect errors and you can correct them. Now, in order to quantify this property of your encoding approach, you could come up with different metrics. Of course, you could, for instance, calculate the percentage of errors that could be introduced into your DNA by means of, in the synthesis process, in the sequencing process, and then quantify the amount of errors that your encoding scheme could handle with under the assumption of, let's say, IID errors, etc. So you could quantify this property, but again, you are failing to take the exact representation of your data into account. Okay, so different encoding approaches will have different data characteristics and will produce different errors. They will most likely not be IID. They will depend on your data, depend on the length of your code word, depend on all kinds of characteristics. So this sort of blurs the true characteristic. So again, we used simulation to get a better idea of this error correction property of our approach. And for instance, here you can see a simple benchmarking setting where you ask yourself how many years can you store your data, and you're able to retrieve the data with a very high probability. Okay, so if you ask this question and you set you clearly define your use case, you can get very good information about how error resistant your encoding method is. A third example where we applied simulation in our project was, of course, parameter optimization. It has a similar idea. The question is, so this is a use case where we use an LT code to error correct our data. And there's a parameter involved in the LT code. And this, of course, affects the redundancy of your data. And it also affects the ability or the probability with which you can correctly retrieve your data. So for instance, if your use case was to save your data for 1,000 years, so that's 10 to the power of 3 years, then you could see that your parameter, which is optimal, is somewhere below 4 and between 1. So you could, obtain a parameter which does not negatively affect your data density, but establishes a good error correction property. Okay, so these were the kinds of questions that we used simulation to tackle or to answer. At the beginning of the project, of course, we went through the literature to see what kind of simulation tools are available. And for two reasons, we decided to start developing our own software from scratch. First of all, this had to do with the fact that we use assembly-based. We assemble short oligos, and the simulation of this kind of synthesis hasn't been, at least to my knowledge, published yet, or how we can simulate this. And second of all, there was a clear lack of software tools, or let's say available software tools that would allow you to do the end-to-end simulation, to really start with the encoding or even with the binarization, and would go all the way to the back. Now there is a preprint of a review article which is on the archive database from last year by Professor Gupta, and it sort of underlines our finding that there's really a lack of simulation tools, and so this supports our feeling. To give you a few examples of the simulation tools that are available, the MESA tool, which is very useful, I think it's developed by the Uni Marburg, and it allows you to simulate the synthesis, storage, PCR, and sequencing, and it gives you many features. So you can clearly, characterize the error patterns. There are different technologies. It has a graphical user interface. So it's very useful. It's open source, but it didn't give us the possibility to do an end-to-end simulation. The same holds true for the DNA storylator. This has been developed by by Eitan Jacobi's group at the Technion. It also includes features for simulating synthesis, storage, and sequencing. But in addition, it also allows you to use different, I don't know if you can read this, but it allows you to use different clustering and reconstruction algorithms at the end of the day. And finally, there's also this DNA Sim product, which I think colleagues have talked about on Wednesday and yesterday, but there's almost no literature about it. It's a commercial software, and we couldn't really find out what the exact features are. So these are the main components of the DNAByte framework that we will publish in the very near future. It consists of 3 main parts. There is a Python module which does the number crunching, which allows you to perform every step in the pipeline. There is a graphical user interface which gives you instant access by means of a browser, and you can upload data and play around with it. And there's also a simulation framework which allows you to answer certain specific questions. Now, here is an illustration of the pipeline of the DNA data storage process. You've all seen this a million times, and that's exactly the point that I'm making here. The pipeline is always the same. So you always start out with some kind of a digital file, you binarize it, you encode it, you simulate the 3 error channels or maybe more, then you do the reverse by correcting and restoring, etc. So what I'm trying to say is that this pipeline is inherently modular, and that you can very easily create good data objects that essentially represent these data characteristics, and create methods which allow you to move from one step to the next. And this modularity, which is inherent to this pipeline allows you to, first of all, create a very modular code, which is in itself a good feature, but it also allows you to be extendable. So the way it's built, the software is built now, is you can just simply add a file to the repository, and you can plug in a new encoding method, or you can plug in a new kind of simulation, but without changing or tweaking the architecture, the skeleton of the software. So you can use basically different combinations of encodings and synthesis methods, and yeah, with very little overhead, with very little programming work, you can create all kinds of different simulation. And here, for instance, you can see the parameter object. So, but just simply changing a single value here, you can modify your simulation and thereby create instant and very easy simulation results. Okay, so as a result of this this work of developing such a simulation framework, we've sort of stated what we believe would be good properties for a standard simulation tool, and we thought that, or we came up with these 5 characteristics. First of all, as I've already mentioned, the DNA simulation framework should be modular and extendable. Now, this is rather easy to do. It should include a benchmarking feature which allows you to compare a certain encoding strategy with others for a predefined use case, and this should be simple to do. We believe it should be open source because the scientific community should be invited to participate, and this should be actually collaborative work. And it also should have some kind of certification. So the results of the simulation should satisfy some criteria which allow you to state with a certain evidence or with certain certainty that the results are trustworthy and are accepted by the community. Okay, so to summarize the key takeaways, benchmarking different DNA data storage approaches requires the use of specific use cases, or the definition of certain use cases, and requires an end-to-end simulation of the entire platform to get good data. We've developed a framework which fits this description and which can solve some of our problems. And I would like to reach out to the community. I think this is the best opportunity for me to reach out to collaborate. If anyone is interested in simulating your methods, if you want to, if you have any needs, then we would be very happy to collaborate with you and take this and develop this tool further. Thank you.
Thank you, Fabian. Are there any questions?
Thank you for your presentation. I'm wondering what model you use to simulate your errors in synthesis, in sequencing. Is it based on learning from experimental data? Is it based from the literature? And especially, can you, since your aim is to biologically produce your code words, what did you have data on the impact of this method of production of your DNA on the error rate?
Okay, so it's patchwork at this moment. So for the sequencing, of course, we use the the methods that we find in the literature. The MESA tool is actually a great resource for these, so we've just basically adopted it for our purposes. Synthesis is not so easy in our case. As I mentioned, we use a block approach, and for the time being, we sort of we simulate this by the user can use concentration of the different oligos, and you can set certain parameters, and eventually you just simulate random interactions between your oligos, and then you can plug in some kind of a in-silico model which gives you a probability of a correct binding. And by this way, we're trying to get a realistic set of double-stranded oligos that includes obviously also incorrect bindings, and that give you the quantitative measure of the efficiency of your reactions. We've heard, I think, by talk from Gamma, that their efficiency with using DNA enzymes is very low, and we had similar experiences with T4 ligase, and at the moment we're at the process where we want to try to align the results from the lab with our simulation to see whether the simulation results actually represent what we find in the gel or what we find in the fragment analyzer. So yeah, we're sort of still figuring out the best model to simulate this block by block synthesis.
Any other questions?
Thank you, Fabian. It was great talk. How about the other synthesis method? How can you simulate those? Because we are also working on the synthesis, but not your method. Does it also work on that?
Yes, so actually we've just stolen from the literature here. There are, in our tool at current, the possibility to just simulate an IDT synthesis by using, I don't know, an array and a column synthesizer, and also Twist has is characterized with an error structure. So we've just implemented the simulation, the error characteristics that were available in literature. But we haven't developed anything new on this.
Just as a comment, I am really willing to do some experiments and try to check the accuracy and also some sort of validate your system before sequencing and after sequencing, and compare it with your simulation.
Totally do that, yes.
Thank you.
Anybody else? Okay, then we'll move on to the next presentation. Thank you.